today I would like to talk about how inbreeding depression in growth shapes competition and fitness. And so that's a work I did at Temple University with my advisor, Rachel Spiegler, and I recently moved at the University of Florida.
Claire, we really can't hear you. Yeah, we're going to need.
Oh, you can't hear me?
Start talking now.
Okay, is that better? No, because I know my voice is, okay, thank you. So mutation continually introduces deleterious mutation. And in inbred individuals, those deleterious mutations, even if they are recessive, are expressed at an homozygous level. Therefore, inbred individuals have a lower fitness than outbred individuals, which generate inbreeding depression. However, the effect of those deleterious mutations on fitness can depend on the trait value of the conspecifics. So here I represent a population with each bar representing a group of individuals from the more inbred on the left to the less inbred on the right. And in individuals with inbred individuals with deleterious mutation have a lower fitness, which are represented by this color gradient from green to blue. And so let's say we have three populations with different level of inbreeding depression. Different level of inbreeding. So if the deleterious mutation affect a trait like the intrinsic growth rate, which is the ability of the plants to grow and flower before the end of the season, individuals with deleterious mutation can't grow fast enough to reproduce before the end of the season, so they will be eliminated. So fitness depends on the absolute trait value and doesn't depend on the conspecifics. And as you can see here, we purge deleterious mutation, and population with a high inbreeding level will have a lower fitness, here null fitness, compared to population with that are more outbred. Now, if selection is soft, so for example, for a trait like competitive ability, which would be the ability of the plants to suppress the growth of their competitors, fitness will depend on the trait value relative to conspecific. So an inbred individual, if it competes with an outbred individual, will not perform well, but if it competes with other individuals carrying more deleterious mutations, then this individual can perform pretty well. So in this case, deleterious mutation tends to be more protected than under art selection, and the population inbreeding level will not affect the population fitness, because as you can see here, each population will contribute to the next generation. And what's important to notice is that hard and soft selection are two extremes of a gradient, and for a trait like competitive ability, the position on this gradient will depend on the strength of competition. So understanding competition is really important, but it's tedious because competition is a dynamic process. So within a generation, individual will germinate, and as they grow, they will start to compete with each other, and competition will increase within the generation. In addition, the type of competitors and the frequency will affect the growth. So there is this nice empirical study from Trinder and collaborators that measured the biomass growth over time for two species. So we have here Dactylis and Plantago. And so they measure growth when the species grow isolated or when they grow together. And as you can see, isolated plants perform better than competing plants. And in addition, if we measured competition at that time, here we would conclude that Plantago is the best competitor, but later in the season, Dactylis is the best competitor. So the timing at which we measure competition is also very important. And to finish, competition is also size dependent. So if we have two individuals with the same size, they will share resources in their overlapping area, and they will each acquire the same fraction of the resources. But if one individual is smaller than the other and competition is size symmetric, the smaller individual will acquire less resources than the big one. Now, if competition is size symmetric, the big individual will acquire disproportionately more resources than the smaller one. So size asymmetric competition increases initial size differences, and this will affect the growth of the individuals and therefore the competitive dynamics. So based on all those information, we are wondering how does the effect of inbreeding depression in growth on the dynamics of competition and population fitness depend on the population rate. And as a secondary question, we also wonder how does the purge of deleterious mutation for growth depend on the population selfing rate. So for this, I use an individual-based model that is spatially explicit. And I will start by showing you how individuals grow and compete, and then I will explain how I introduced inbreeding depression. So I use a zone of influence model, ZOI model. This is a semi-mechanistic and conceptually simple model, so it makes it very easy to test a prediction. And so each individual plant is associated with a disk. It's a zone of its zone of influence within which it will acquire resources. So the bigger the ZOI, the more resources the plant can acquire and therefore convert those resources into growth. But if there is competition, the amount of resources acquired is decreased and therefore growth is decreased. So here we model the growth of an annual plant with shoots and roots growing differently. So we have ZOI for the roots and another one for the shoots. And the growth of an individual will be proportional to the intrinsic growth rate, multiplying a factor that is the difference between the smaller value among the ZOI for the shoots and the roots, and the ratio of the square total biomass divided by something proportional to the maximal biomass. So in this growth model, we have three K parameters: initial size, B naught, intrinsic growth rate, R, and maximal biomass, B max. And for each of those growth parameters, we can have inter-individual variation around the mean, just to represent phenotypic variation. And as plants grow, their zone of influence will overlap. And so in this overlapping area, plants will have to share resources, so they won't acquire all the resources in their zone of influence, but only a fraction, which will define the effective zone of influence. And so growth will be affected by this zone of this effective zone of influence. So competition decreases growth. And if plants can't acquire more than 1% of the resources within their ZOI, they can't maintain themselves and die. So that was the general model. And now, for the first time, we introduced inbreeding depression in this model by decreasing the parameter values for inbred individuals. We assume the same inbreeding depression for all self individuals, and we compared population with different selfing rates from fully outcrossed to fully inbred. And we perform simulation on the NetLogo platform. Each population starts with 100 individuals that are randomly located in the plot, and we replicated each simulation 10 times. For the results, I will explain the effects of inbreeding depression on each growth parameters, and then on when we have inbreeding depression on all of them. So for inbreeding depression on initial size, B naught, I'm showing the growth of an isolated plant. So we have the biomass over time for an outcrossed individual in black and a self individual in blue. And as you can see, there is hardly any difference in their growth. So just to save time, I'm just going to tell you when plants compete in population with different selfing rate, we did not see any effect on growth and on population fitness. So I'm skipping this part. Now with inbreeding depression on the intrinsic growth rate, I represent the average growth by the thick line and some inter-individual variation by the less opaque curves. And you can see that self individuals are expected to grow smaller than outcrossed plants. And when we look at the growth within competing population, we can measure the proportion of resources acquired through shoots for different selfing rates. And we see that indeed self individuals, the open symbols, acquire less resources than the outcrossed plants. And as the selfing rate increases, the frequency of self-plants, so the small plants, increases, so competition decreases, and the fitness of both self and outcrossed individual can increase. We can also look for one selfing rate at the temporal dynamic, and then we see that the proportion of resources decreases over time, so it means that competition increases over time. For mean individual fitness that I measured as the product of survival and fecundity, with fecundity being proportional to the shoot biomass, we see a similar pattern with self plants having a smaller mean individual fitness than the outbred plants, and an increase when the population selfing rate increases. So here you can see that the difference in mean fitness between self and outcrossed decreases when we increase the population selfing rates. So meaning that we have an interaction between the mean individual fitness and the population selfing rate, and this is a benchmark of soft selection. We also see a difference in the fitness of outcrossed plants in pure outcrossed population and the fitness of self plants in pure self population, which indicates some hard selection. When we look at population fitness, it decreases over the range of selfing rate, indicated that hard selection is stronger than soft selection. And for effective inbreeding depression that I define as relative difference in fitness between outcrossed and self individuals, we see that it's relatively strong and stronger than the initial inbreeding depression that we applied on the growth parameters that I set at 0.4. And in addition, it decreases as we increase the population selfing rate, because as you see here, the difference tends to decrease. So which indicates that we have soft selection again, but that deleterious mutation are may be more protected at high selfing rates than at lower selfing rate. For inbreeding depression in Bmax, we expect differences in growth only later in the season. And when we look at the temporal dynamic for a population with a selfing rate of 0.6, this is indeed what we see. Initially, they grow the same, and later in the season, you can see the differences in resource acquisition. And because those differences emerge later in the season, it means they have less time. There is less time for differences to accumulate. So that's why you can see a difference among self and outcrossed individuals that is smaller than with inbreeding depression in R. And this pattern is consistent over the range of selfing rate. Here's a smaller resource acquisition for the self individuals and for the outcrossed translate into lower mean fitness for the self than for the outcrossed. And in addition, because the differences in resource acquisition emerged later, there is less mortality for the self plants than for the outcrossed plants. Sorry, there is less mortality for the self plants than with inbreeding depression in R, and more mortality for the outcrossed plants than with inbreeding depression on R. So that's why the fitness is better for the self plants and lower for the outcrossed plants compared to inbreeding depression in R. And here we don't really see a big difference. I mean, the difference in mean fitness among self and outcrossed individuals doesn't really change with the population selfing rate. So it means that if there is soft selection, it's probably very weak. We again observe some hard selection on Bmax. And again, the population selfing rate decreases when we increase population selfing rate, showing that hard selection is stronger than soft selection. Effective inbreeding depression is lower than with inbreeding depression on R, because here again, the differences among self and outcrossed individuals in mean fitness are lower. And now when we have inbreeding depression on all growth parameters, we expect self plants to grow smaller than outcrossed plants, with early differences being controlled by inbreeding depression on R and on Bmax, and later differences being mostly controlled by inbreeding depression on Bmax. When we look at the proportion of resources acquired through the shoots, for the outcrust and the seft, you can see that it's almost exactly the same to what we observed with inbreeding depression in R. And this is consistent over the range of selfing rates. So it means that the early differences we observed in growth accumulate and probably overweight the later differences in Bmax. And despite the survivor acquire the same amount of resources, inbreeding depression on all growth parameters increases mortality compared to inbreeding depression on R. So the fitness of the self-plant is smaller than previously with inbreeding depression on R. The fitness of the outcrust is approximately the same. So again, we have soft selection and we observed hard selection. And the higher mortality of the self-plants translates into a more dramatic decrease in population fitness when we increase the selfing rate. Effective inbreeding depression is stronger, but still we have this decrease in effective inbreeding depression when we increase the selfing rate, showing that high selfing rate purge less deleterious mutation. So to your first question, we can answer yes, the effective inbreeding depression in growth on the dynamics, the effect of inbreeding depression in growth on the dynamics of competition and population fitness depends on the population selfing rate. In addition, we showed that inbreeding depression on growth parameters like B0 will have no effect on growth curves and therefore on competition dynamics, but inbreeding depression on R will have a strong effect on the growth curve and therefore on competition dynamics. And we also showed that population fitness decreases with selfing rate, suggesting that selection is harder than soft. And to your second question, we can also say that yes, the purge of deleterious mutation for growth depends on the population selfing rate. We found strong selection against deleterious mutation, but population with a higher selfing rate, so where competition is lower, can maintain more deleterious mutations than population with a low selfing rate. So thank you very much for your attention. I also would like to thank the Society for funding this talk, and I would be more than happy to take any question or comment you would have.
You had a power of 4/3 in your differential equation,
Mm-hmm.
and I'm curious if there's any intuition for a little exponent like that.
Yeah, that's a good question. So that's a growth model that I just used that was already in the literature. And I think they compare to empirical data, so maybe that was to fit better to what they observed. Oh.
Like an allometric kind of.
Yes, yes.
Oh, yeah. So it's a spatial model, but I wasn't clear how the spatial pattern affects the outcome. Do you assume some sort of average random distribution, or are they clustering?
Yeah, so they are randomly located. So by chance, some plants will just be alone, at least at the beginning, and some plants will be clustered together. And so that's why we have some replication to take into.
Does this propagate through the generations, or?
So it's only one generation. I'm looking at what's happening within the growing season.